Hey, welcome back. It's Miss Ramon. This is part 2 of the review for the Unit 7 test for Summer Parish Virtual Honors Bio. And we will begin with a discussion of meiosis. And for test questions for meiosis in general, those questions will be similar to the ones for mitosis in terms of there's going to be vocabulary, factual questions about the names of the stages of meiosis, what's happening in each of the stages. Can you recognize a image of particular stages in meiosis? Are the chromosomes duplicated? Are they unduplicated? Is the cell haploid? Is it diploid? All of those types of questions again. But now that you know mitosis exists, you're going to have questions that compare and contrast mitosis and meiosis. And then meiosis has 2 overall stages, so you'll need to know how meiosis in general compares with mitosis, but then also how meiosis 1 compares to mitosis, how meiosis 2 compares to mitosis, and how meiosis 1 and meiosis 2 compare to each other. And then also added in this material are the concept of linked genes, and the law of independent assortment and the law of segregation. All right, let's start with the general facts of meiosis. Meiosis is or does have 2 stages. So it goes through prophase, metaphase, anaphase, and telophase twice, and meiosis is called the reduction division because the cell, after the division, will have half as many chromosomes as it did at the beginning. And these images come from the Khan Academy. You know that I love their images. And important facts or important things to look for when you're studying this material. Always ask yourself, is the cell haploid or diploid? Are the chromosomes duplicated or unduplicated? And by asking yourself those 2 questions, it will help you determine, is this meiosis or mitosis? And then, is this meiosis 1 or meiosis 2? And I have highlighted in yellow the answers to those questions for meiosis 1. If you notice prophase 1, it looks just like prophase of mitosis. However, there are some events that happen in prophase 1 that don't happen anywhere else. It doesn't happen in prophase 2, and it doesn't happen in prophase, and that is the formation of what's called the tetrad. So let's look here at the tetrad. I don't know what my dog is trying. Choco, you're on a pillow on the couch. That should be the most comfortable place. For some reason, sitting on top of the laptop looks more comfortable than the pillow. Baby, I love you, but look, sweetheart, look at this pillow. It's so, I'm like trying to do the video, and then I'm like not hitting, I never hurt my angel, but I'm like pushing her away. Look, now, see, you're like the princess and the pea. You've got all these pillows to lay on. There's a good girl. Let's see if that works. Okay, once again, when there's a pandemic and mommy has to make these videos at home, I have to work at home. I don't have a choice. All right, back. No, okay, seriously? Are you kidding me? No. So I have a picture here of the tetrad, The tetrad is when a homologous pair is in synapsis. And synapsis is the description of what happens for a tetrad to form. So it's sort of circular definitions that the tetrad forms by synapsis, and synapsis is what forms the tetrad. The tetrad is the structure itself. So when you're talking about the tetrad, you are talking about this homologous pair that are quote unquote on top of each other. And because they are on top of each other, crossing over can occur. Your DNA actually can break off and reform quite easily, which is frightening to me. And when the chromosomes are in synapsis, so when they're being held together, they are in proximity that they can break off and reform and then actually swap chunks of chromosome with each other. And we'll talk about the significance of that in just a moment. But right now, what's important is that the tetrad forms during prophase 1. And that happens by the process of synapsis, or you can think of it that way. Synapsis is what holds the tetrad together. When you take more difficult biology courses in your life, if you do, you'll learn a better way to describe that. But for this class, synapsis holds the tetrad together, and the tetrad is constructed of a homologous pair. And it begins in prophase 1, and it does not occur during any other stage. The tetrad will still be together during metaphase 1. So the tetrad is present in metaphase 1, but the tetrad formed in prophase 1. So you have to be careful of what the question asks you. If the question asks you, when does the tetrad form, prophase 1 is the correct answer. Metaphase 1 is not the correct answer because the tetrad was formed before metaphase 1. So that's why that answer would be incorrect. But if you're asked, when is the tetrad present, then it would be prophase 1 and metaphase 1. So let's take a moment just to look at crossing over in a little more detail, because crossing over is significant, not just when you're learning about meiosis, but also the consequences on evolution. The tetrad formation does impact natural selection. And the reason is, remember that in natural selection, because organisms are born with a range of traits, if the environment changes, if the environment changes, then some traits might be beneficial and others might not be beneficial. So for a species to have this range of traits with each generation, it improves the fitness of organisms if you are continuously producing all these new traits and range of traits, because if the environment changes, some of these new traits or new combinations of traits might be an adaptation that maybe wasn't an adaptation before. And crossing over is beneficial to increase the different traits or the different range of traits, because when crossing over occurs, it creates a new combination of traits that were not there before. So what that looks like, I've made this cartoon. We have a lady, and she falls in love with a man, and they get married, and they have children. And they met at work. You see, they're at work. And the mom has blonde hair, blue eyes. She's right-handed, as you can see. And she has her second toe on her foot is longer than her big toe. Her earlobes are not attached. And then the love of her life has black hair and black eyes. He's right-handed. His second toe is shorter than his big toe, and his earlobes are unattached. So they have all these different traits, and some of the traits are on the same chromosome. And traits or genes that are on the same chromosome are called linked genes. So for this couple, the eye color, the hair color, and their handedness are on a chromosome together. But the big toe being longer than the second toe being longer than the big toe, and the attached earlobes are on a different chromosome. So I've just decided that hair color, eye color, and handedness is on chromosome 12. And then the attached earlobes and the second toe length, let's say, are on chromosome number 13. So when they pass their traits on to the next generation, mom will pass on hair color, eye color, and handedness on one chromosome, and then dad will hand down those traits on his chromosome 12. So they have a son, and when their son is born, he gets one of his chromosomes from mom, from her egg, and then he gets one chromosome from his dad, from his sperm, and then that creates our diploid child that has 23 pairs of chromosomes, and one set is from mom and one set is from dad. So when mom and dad pass their traits on, those traits that are linked, that are on the same chromosome, will be inherited together. You are only able to inherit those 3 traits on that same chromosome together. So our child has inherited the 2 copies of the chromosomes, and the way that those chromosomes interact. He has black hair, black eyes, and he's left-handed. And that chromosome from his dad is the dominant trait. If he shows one, he's showing all of them. And of course, what I'm saying is not 100% true. We'll learn about Mendelian genetics and inheritance patterns on the next test. But for this information, we are going to, we're just going to pretend that those are the dominant traits. But the point is, the chromosome he inherits from his dad has on it black hair, black eyes, and left-handedness on that chromosome together. Mom's chromosome, he inherited together the combination of blonde hair, blue eyes, and right-handedness. Now, when he makes his gametes, this will be during meiosis. So during meiosis, the chromosomes duplicate, and then during prophase 1, we have this opportunity for crossing over. So by the end of meiosis 2, his sperm, his gametes, now include combinations that weren't there before. So all of your homologous pairs separate from each other and end up in their own gametes. And the chromosomes that he inherited from his parents, you see that some of the gametes will have exactly what he inherited. But because of crossing over, we now have these chromosomes that have a new combination of traits. It's not that it's necessarily a new trait. So he is not passing on green eyes or red hair. It's still blue eyes or black eyes, black hair or blonde hair, left-handed or right-handed. It's still those traits. But now we have a chromosome that has a combination of traits that weren't there before on one chromosome. We hadn't seen before black hair, black eyes, and right-handedness on one chromosome together. We had seen black hair, black eyes, and left-handedness together. We had seen blonde hair, blue eyes, and right-handedness on a chromosome together. But now, because of crossing over, these two chromosomes on the right, these combinations are new. He is now passing on a combination of traits that weren't a combination of traits before. And that's what makes crossing over so significant, and why there's a lot of emphasis put on crossing over when you first learn it. The implications it has on natural selection is what's significant. A new combination of traits that were already present, they just weren't linked before. So that influences the traits of the next generation. And then the offspring could also have their chromosomes undergo crossing over, and that would make an even different combination of traits. So that is why crossing over when the tetrad forms is so important. It's not that new traits arise, it's a new combination of traits that are inherited together. This one chromosome will be in sperm. So if these are the 4 gametes produced by meiosis, there's an equal chance that any of these gametes will fertilize an egg, and then now we have offspring that have a new combination of traits that weren't together before. All right, so let's go back to meiosis 1. What you're looking for, questions you'll be asked. You will be given one of these cells with nothing else, and then you could be asked, is this meiosis or mitosis? Is this meiosis 1 or meiosis 2? Ask yourself, is it haploid or is it diploid? Is it duplicated or unduplicated? Notice that in meiosis 1, the chromosomes are duplicated the entire time. So if the chromosomes are unduplicated at all, then you can just disregard meiosis 1. It's not going to be meiosis 1. Notice that during meiosis 1, the metaphase 1 stage, the tetrad lines up together. And that means during anaphase 1, the homologous pairs are separated, and also the tetrad is broken apart. The tetrad does not exist in anaphase 1. The homologous pairs are there, but not the tetrad. Homologous pairs and tetrad do not mean the same thing. Tetrad is this formation with synapsis. Crossing over is only possible while the tetrad is formed. So crossing over can only happen during synapsis. That's an application question because synapsis is only when is only when the tetrad has formed. And remember that the location of crossing over is called the chiasmata. So that's where the crossing over occurs. And anaphase 1 is the separation of the homologous pair. And then during telophase 1, the cells are becoming haploid. In mitosis, the cells are diploid the entire time. During meiosis, we will begin with a cell that's diploid, but we'll end with a cell that's haploid. In meiosis, the cell goes from diploid to haploid pretty quickly. Most of meiosis is the cells are most, not meaning like 99%, but a majority of more than 50% of the time, let's say, the cell is going to be haploid. So when you're looking to see is it diploid or is it haploid, if the cell is diploid, then it cannot be meiosis 2. Meiosis 2 begins and ends haploid. And during meiosis 1, the chromosomes are duplicated the whole entire time. All right, now meiosis 2. Meiosis 2, as I said earlier, begins and ends haploid. And the chromosomes are duplicated during prophase 2 and metaphase 2. Anaphase 2, the sister chromatids separate. And that's what happens in anaphase for mitosis. But the difference is the anaphase and mitosis is diploid. Mitosis is diploid from beginning to end. And meiosis 2 begins haploid and ends haploid. So the way to tell anaphase 2 from anaphase and mitosis is to look to see is the cell diploid or haploid. Anaphase 1, the homologous pairs are separated. So it looks very different from anaphase 2 and from anaphase. So anaphase 2 and telophase 2, we have unduplicated chromosomes, and it's haploid. So looking at all of the cells during mitosis, meiosis 1 and meiosis 2, those are questions you'll ask yourself to determine what stage the cell is in and which process the cell is in. Then once you know what stage and what process, you can also expect questions that address what is the location for the centrosomes. Where or is there a nuclear envelope? So those kinds of questions you can answer once you know what stage of the cycle the cell is. And now that I've pointed that out, let's go back to meiosis 1. So in meiosis 1, the homologous pairs form tetrads in prophase 1, line up together in metaphase 1. Anaphase 1, the tetrad is broken, the homologous pairs are separated. By the end of telophase 1, the homologous pairs have been separated from each other. They're in totally different cells. So now the cells are haploid. A diploid cell would have the homologous pair. At the end of telophase 1, there are not homologous pairs anymore in these daughter cells. And also, after telophase and cytokinesis 1, we now have just the one centrosome present in each cell, and our nuclear envelopes have reformed. During meiosis 2, the second centrosome will be synthesized and then the centrosomes will move to the opposite ends of the cells. Our cells are haploid. When the chromosomes line up on the metaphase 2 plate, we don't have homologous pairs. It's haploid. The sister chromatids are separated in anaphase 2. The cell is still kind of considered haploid. And then by the end of telophase 2, we have our haploid, unduplicated chromosomes in the nuclear envelope, which has reformed. And then our 4 daughter cells, each are haploid with unduplicated chromosomes. They decondense, and each cell only has the one set of centrioles, so they have the one centrosome, and they won't construct a second centrosome. Actually, for these cells, since they're gametes, they won't divide anymore, so they will not undergo any kind of division after they are created. Now, I have taken metaphase and anaphase cells from each of the types of division, so you can see those differences. So ask yourself, is it diploid, is it haploid? Are the chromosomes duplicated or are they unduplicated? So looking at these 3 cells, metaphase from mitosis, we see that it's diploid. There are 2 long chromosomes and 2 short chromosomes, and that's what you're looking for to determine, is this cell diploid or haploid? Do we have 2 long, 2 medium, 2 short? You're looking for lengths. So this is a diploid cell, but there's no tetrad. Look at metaphase 1. This is a diploid cell, but it has tetrads. And if we look at metaphase 2, this is not diploid. There's 1 long and 1 short chromosome. So if it's haploid, it has to be metaphase 2. If it is diploid, what you're looking for is the single file line or the tetrad. Single file line means mitosis. Tetrad means meiosis 1. Now looking at anaphase, sister chromatids are being separated in anaphase of meiosis. Sister chromatids are being separated in anaphase 2 of meiosis 2. But notice that anaphase of mitosis is diploid, and anaphase 2 of meiosis 2 is haploid. That's the difference. That's what you're looking for. In anaphase, in mitosis and anaphase 2, these daughter chromosomes are unduplicated. So that means it's either mitosis or anaphase 2. Anaphase 1, we still have duplicated chromosomes. And anaphase 1 does have the duplicated chromosomes because it's the tetrad that is separated and not the sister chromatids. So that makes this anaphase 1. So knowing which trait goes with which type of division, you'll be able to figure out which type of division it is. Is it diploid or is it haploid? Are the chromosomes duplicated or are they unduplicated? You could get questions where you have the pictures of the cells. You also might get questions that's a word problem, and you may want to draw out what that cell looks like when you're determining the answer. If you're a really visual learner like I am, I would have to draw it out. But while we're looking at these, let's practice chromosome number. So remember, chromosome number is the number of chromosomes that you see in the cell. So for our metaphase cell, the chromosome number would be 4. And the chromosome number for metaphase 1 would also be 4. The chromosome number for metaphase 2 would be 2. There are only 2 chromosomes there. All right, now let's look at our anaphase cells. The cell in anaphase, the chromosome number is 8. There are 8 chromosomes here. When the sister chromatids are pulled apart, they are now considered chromosomes. So at this moment, there are 8 chromosomes, and it won't be 4 chromosomes until telophase has completed. In anaphase 1, our chromosome number is 4, just like it was in metaphase 1. In anaphase 2, the chromosome number is 4. And that's because the sister chromatids, once they're separated, are now considered chromosomes. So that would be actually a really good application question, because during mitosis, the chromosome number doubles between metaphase and anaphase. And the chromosome number doubles again in meiosis 2. Between metaphase 2 and anaphase 2, we go from 2 to 4. In metaphase, we went from 4 to 8. But the chromosome number stays the same between metaphase 1 and anaphase 1. It's 4 and it's 4. So, that makes a really good application question. I'll have to think about that. All right. We will stop this video here since it's been 30 minutes, and then I'll pick back up with a discussion about linked genes and how to determine if genes are linked or not. All right, I'll see y'all in part 3.